Hi, my name is Emily Stene. I'm the Childhood Cancer Survivor Program Nurse here at the University of Minnesota in the Department of Hematology Oncology in Pediatrics. I'll be moderating this session for you today with our wonderful speaker, Dr. Abigail Johnson. Dr. Johnson is an assistant professor and registered dietitian in the Division of Epidemiology and Community Health in the School of Public Health. She's also Associate Director of the Nutrition Coordinating Center. Dr. Johnson has diverse experiences in nutrition research, and her present research explores the relationships between diet and the human GI tract microbiome in health and disease, specifically in prediabetes and cancer. We were supposed to have a patient perspective for this session as well, which you'll see on your agenda, but we have had to pivot a little bit, and we will be not offering that for this session. Before we kick things off, I wanted to share a few ground rules for the session. The goal of this session is to help those in attendance feel better, more hopeful, and not so alone. We're not going to talk about specific diagnosis or treatments or give medical advice specifically, and we will not be discussing side effect management. If you're joining us online, our in-room moderator is Kiara, and she'll be following along with your comments, so feel free to chat in supportive comments and related questions into the chat. We'll have time at the end of this session for a Q&A, and with that, Dr. Johnson, you can take it away.
So my name is Abby Johnson, and I am, like she said, an assistant professor here in Epi and Community Health, and I'm trained as a registered dietitian and a nutrition scientist, and my research interests are firmly in the area of gut microbiome and diet. So as a show of hands, who has heard of the gut microbiome? Almost everyone. Who could tell me what the gut microbiome is? We got one. Yes. So my hope for you today is that after this talk, you'll have a better idea of what the gut microbiome is, what we mean when we say that, what that means when you read it in a news article that says the gut microbiome is causing this or is linked to that, and that I hope that I'll be able to explain to you a little bit more about how we look at microbiome data. We'll do a little activity where you'll put your scientist hat on and you'll be able to understand a little bit better how we come up with some of the analyses that we do. And I'm going to show you a little bit of data from my research. I hope it won't be too overwhelming, but I do love data, and I think that it'll be helpful for you to see where some of what we say comes from in terms of what we're actually drawing conclusions from in terms of the data. And then I will end with some practical recommendations for feeding your gut microbiome. So, this is one of my favorite pictures in science communication. This is an image that was published in the National Geographic. It's a false-colored electron micrograph of the microbiome. So what I love about this picture is that you can really get a feel for the complexity of the microscopic environment that's inside of the gut. I think this is from a mouse gut. And what you can see is these purple fungal hyphae that extend across the screen. You can also see lots of blue and green small cells, and those are bacteria in the gut microbiome. So we can define the human gut microbiome as the community of microorganisms, and this is typically bacteria, fungi, and archaea that live within the gastrointestinal tract. And often what we're really talking about is poop. We are not easily able to sample the microbes that are in the colon in other parts of the colon. So what we rely upon are the microbes that you excrete daily. What I love as a nutrition scientist, as a registered dietitian in this picture, is that running diagonally through the image is what looks like a piece of tree bark, right? It looks like a felled log, and that is fiber shown at the microscopic level. And so you can really appreciate here how the bacteria in the gut are spilling over and interacting really closely with the remnants of digestion, with the leftovers of the food that you consume when it reaches the large intestine. Okay, so how do we get to the point that we can say anything about this complex community? The challenge with the microbiome is we can't do what microbiologists have typically done throughout all of their time as scientists, where they've picked a bacterium and they've put it on a petri dish and they've grown it up in a single culture. So they have lots of cells of the same species. We have complex communities in our guts that are thousands of strains of different species, and they can't all be grown in isolation. We're getting better at isolating and growing different bacterial species, but we can't grow them all. So how do we know who's there if we can't grow everything? We use sequencing to work out which bacteria are in our gut microbiome. So what we do is we take a sample and we extract the DNA from that sample, and we use robots, typically, to put all of the samples that we have onto plates, and then we put those into a sequencer. And what we get out looks like this. So that's the microbiome. That's actually my microbiome on the screen, or a fraction of it, because this file goes on and on and on and on and on and on and on. These files are enormous, and they have to live on the supercomputer. If you tried to download them to your laptop, you would run out of room. So each line on this file, or every other line on this file, is the actual DNA sequence from a part of the DNA from a microbe in the gut. This is kind of a black box of sorts, right? Like, how do you figure out who's there from that mess, right? That's a lot of information. And then we get to telling you something about the different species that live in your gut. So, refresher, take yourself back to maybe sophomore year high school biology. You probably learned about the kingdom, phylum, class, annotation. This is the example for E. coli bacteria. We typically look at bacteria in the microbiome, often at the genus level and sometimes at the species level, and then occasionally at the strain level, which I don't even have on here. So, to help you understand this, I'm going to walk you through an exercise that I do with my students where we use jelly beans to understand sequence alignment, because I think this is a complicated idea, but it underpins everything that I'm talking about in terms of like who's in your gut microbiome. So how can jelly beans help us to understand the gut microbiome? Well, just like the microbiome, jelly beans have databases. We have databases of bacterial genomes that help us to align the sequences of the sequences that we get out of a fecal sample to a known bacteria. So in the terms of Jelly Belly jelly beans, we have different flavors of the jelly bean, and we can figure out the flavor based on the color of the jelly bean. So how does classification work in this jelly bean space? We can visually look at our beans, and we can put them into a color category. You'll notice that some will be a little harder to actually put into categories. When I do this with my students, I hand out packets of Jelly Belly jelly beans, and I have them try to categorize them and match them up. And sometimes you can't figure out, like if it's a mottled yellow and green jelly bean, it could be one flavor or another, and you're not entirely sure. And so that's actually true in microbiome science too. Sometimes we don't really know, and so we put things at a higher classification level because we can't figure out what the strain is or what the species is. So we assign them to a color category, right, like this. And then we can count up how many of each of the colors that we have, and we get something that looks like this, where it's mostly red, some yellow, some orange, some green. And then we can make a plot that looks like this, where you have, this is a relative abundance stacked bar chart, which is a favorite visualization of microbiome scientists. You see these everywhere. They're not super helpful in terms of actually what you can see and understand from them, but they are visually pleasing to look at. And if we did this for different samples of bacteria, or in this case, samples of jelly beans, we can get different types of compositions, right? Different numbers of blues versus reds versus yellows versus pinks. And that's what that might look like if you had 3 different compositions. Okay, so back to real data. We have microbiome sequences. We make these bar plots of them. And this is what this looks like for a real study. So this is the Human Microbiome Project data. This was an NIH funded study that aimed to understand the complexity of the microbiome and to try to define what healthy, normal microbiomes look like. And so they took incredibly healthy people and they took samples of their microbiome from a number of different body sites. They sampled their nose, they sampled their mouth, they sampled their tongues, and they sampled their stool. And we'll zoom in on the stool because that's the area that I spend most of my research on. But what you can see from the stool samples, and now that you're experts in understanding alignment and sequencing, you can appreciate that there's not a single microbiome type here that is consistent across all of the people. If there were, this would just be a block of blue, green, and it would be consistent across everyone. What you can see is that in these objectively healthy people, some people have microbiomes that are mostly Bacteroidetes, all blue, and some people have microbiomes that are dominated by Firmicutes, which is all green. But there's not one microbiome type that defines healthy. And this is a challenge in our space of microbiome research, because when people want to shift their microbiome, they want to make their microbiome better and healthier, we don't know where we're pointing them to. We don't know where we're trying to get them because we don't have one healthy. What we do know is that there are clear markers of an unhealthy microbiome. So in some research that I do with some researchers at Memorial Kettering Sloan Cancer Center, we have followed people as they've undergone bone marrow transplants for hematopoietic stem cell cancers. And we see that when they are in treatment and they're undergoing chemotherapy and they're undergoing massive antibiotic regimens, their microbiomes become dominated by single species or just a couple of species. And that's objectively not a great microbiome to have. And so returning to something that's more diverse is often the goal. And what we see when we look across populations is that higher microbiome diversity, so the number of different types of species that you have, is often associated with health and healthful outcomes and healthy behaviors. So we think that looking at diversity, overall diversity, is one of the ways that we can measure what a healthy microbiome looks like, although it's not perfect. Okay, so you might be thinking, where does my microbiome come from? Where do I get microbes? We're filled with microbes. We have microbes on us and in us. Where do they come from? Your microbiome develops throughout the life course. So from the time that you're born, up and through adulthood, and then as you age, your microbiome is sort of changing slowly over time, sometimes more quickly. So during the first 1 to 4 months, your microbiome develops a lot of diversity, and that increase in diversity around 4 months is correlated with the time that babies start exploring their world. They start crawling, they start putting their hands on surfaces into their mouth, and they also start consuming food. So we see that the introduction of complementary foods, so moving from breast milk or formula to eating fruits and vegetables, changes the composition of the microbiome, and along with food and environmental exposures comes more diversity of the microbiome. We know that in adulthood, when people undergo major stressors, when they move, when they experience big changes in health, when they undergo antibiotics, you can see shifts in the gut microbiome. And typically those return back to normal when they are return from their travels or when they recover from antibiotics, but sometimes those shifts cause changes that persist. So why do we care about the gut microbiome? There's been a lot of nutrition research for the last couple hundred years where we've tried to understand how the foods that we eat affect health outcomes, right? So if you think about consuming like a pastry for breakfast, that you eat it, the glucose is released by enzymes from your saliva and through your gastrointestinal tract. You get a spike of blood glucose, and that blood glucose might contribute to how your body manages that blood glucose, might contribute to whether or not you go on to develop diabetes or contribute to if you have symptoms of your diabetes particularly. So we've looked at food to outcomes for a long time. But what we see when we look across populations is not everyone responds to the same foods the same way, and we haven't been able to prove that those differences are rooted in our genetic differences. So I showed you the picture of the Human Microbiome Project gut microbiomes, and there's a variety of microbiomes. So one of the hypotheses we have is that the differences in the gut microbiome may explain why some people respond to different diets differently. because the microbes can do parts of the metabolism of food that we as humans are not able to do, and that the metabolites that microbes can produce could be impacting our health as well. And a lot of the research in this space has mostly focused on things like weight gain or loss, blood sugar control, intestinal symptoms, and inflammation. There's not a lot of research yet in really specific diseases or conditions, in part because this is a relatively young field. It's really only existed in its current form for like 10 or 15 years, and so there's still a ton of research to be done. Okay, so when I joined a lab as a postdoc, so I did my PhD, and then I went and worked in industry and was working as a registered dietitian in the food industry, helping to make like enteral nutrition products, so products that you might consume while you're in the hospital and you're unable to eat food, or things like Boost or Ensure, right? So while I was working in that industry, we often got this question like, how can we change the microbiome with food? And what I realized more and more was that there just wasn't any like literature or research that we could use to really consistently have answers to that question. And so this was the question that drove my return to academia and to doing a postdoc, was what happens to the gut microbiome? Does it actually change with food? How do you change it? What do you tell somebody? Can you tell someone, eat an apple and an apple will change your microbiome in this way? Or eat an orange and an orange will change your microbiome in this way? And we just didn't have answers for that question. And spoiler alert, I'll tell you, we still don't. But we may be a little bit closer. So one of the studies that we did was we asked 34 people to give us microbiome samples every day. And this is a lot of information in a relative abundance stack bar plot, which you're now all experts in understanding. But I'll zoom in on one of them. So this is the microbiome from day to day for this one person. They provided a fecal sample every day for 17 days, and then we sequenced that fecal sample, and we got this information out. And what I take away from this plot, and maybe you can see too and appreciate, is that it's pretty much like bands of red, bands of green, bands of purple. There's not a lot of change from day to day. The microbiome that this person has stays relatively stable. And they look like themselves each day, essentially. And this is another way to plot that information. It's a little overwhelming, but essentially on this plot, it's the same exact information. This time that person is in the blue, dark blue circles, and each of their days that they gave a fecal sample are all colored dark blue. And you can see that their samples are with all of their other samples. And so points on this plot that are close together are more similar to each other than points that are in different, further away points. And what you can also see is that there are people in this study who have very different, distinctly different microbiomes that have totally different members within their communities. And that's the same sort of finding that we saw in that Human Microbiome Project, where you can have healthy people with very different gut microbiomes. But what we found is that people look like themselves from day to day. Now, for the same time period, we also asked people to tell us everything that they ate. Now, raise of hands if you've ever tried to track your diet. So you've opened MyFitnessPal, or you've used Chronometer and you've jotted down everything that you ate in a notebook or something like that. Was it easy? Did you like doing it? No, I'm seeing lots of no's and no. Same is true of my research participants. It was easier for them to give me a fecal sample every day than it was for them to tell me what they ate. They hated it. It was like pulling teeth. You had to call them up every day and be like, hey, I noticed you haven't entered your food into your record. Could you please enter your food into your record? So it's not easy to do. It's actually really challenging to record everything that you eat and drink. But that's what we had them do. And what you might not be able to see from this view, but if I zoom in on that same person, is that when we look at the food that a person eats over the same time period as we have for their microbiome samples, is their diet is much more variable. It bounces around. It's lots of peaks and valleys. And this is actually at the highest level of food classification that I can do. So I've lumped things together as like grains, meats, dairy. They're big, broad groups that encompass a lot of different foods, and it looks very variable. This person's, you can see, in the kind of sandy green color. I don't know if I have a pointer here. These are eggs, and they eat them on the weekend. So you can see them popping up when it's a weekend day. But if I try to view this as a each individual food is given a different color. It looks like Picasso drew this, and we can't really learn anything. So my dream of being able to go apples equals this is totally foiled by the fact that nobody eats the same thing every day from day to day, and I can't make heads or tails of it. And if we look across all of the people in our study, this is another way to look at the variety of foods that you eat. Each white dot is a person, and they are connected to the food that they eat by lines. And the foods in the middle are things that are eaten by most people in the study. So in this study, this was done here in the Twin Cities. This ended up being things like white bread, chicken breast, iceberg lettuce, beer, wine, milk, things that people eat routinely, coffee. They came up and showed up in the center of the pot. On the periphery, these are things that are just connected to just one or two people. And these were more unique things. These were fruits that were not commonly consumed. These were things like organ meats. That type of food ended up on the periphery, but you can see that like almost half of the foods that people report were only reported by one or two people. So it's really difficult to draw conclusions at the food level. Usually in nutrition, we get around this kind of messy complexity in dietary data. Like, think about it. You walk into the supermarket, there's 19,000 foods to pick from, and you're all making different choices, and you're all eating different things. You probably eat differently from people in your own household, right? Even when given the same food offerings, like this morning at the buffet, we all picked slightly different options. So to deal with this messiness, nutrition scientists have historically gone to macronutrients and micronutrients. So have you ever thought about the carbohydrates in your food? Have you ever tried to eat a high-protein diet, right? If you've ever tried to reduce the fat in your food, then you've spent time looking at nutrition facts panel labels and trying to understand, okay, foods are all different, but they all contain sort of these same basic building blocks, and therefore I can change my diet by thinking about those building blocks. And that's what looks like this nutrition facts panel. So this is the facts panel for a strawberry, and strawberries have about 50 calories per serving. Most of that is coming from carbohydrate, and they have some sugars and some fiber, right? Seems pretty simple. They're not a very complicated food. Now, think about being a microbe and running into a strawberry. What do you see as a microbe? Go back to that mental schema, that picture we showed you at the very beginning. What does a microbe see when they see a strawberry? Well, it's much more complicated than what we see on a nutrition facts panel. The macro and micronutrients that we extract from the food are consumed by us, the host, and then the leftovers, the stuff that we don't have enzymes to break down, is what makes it to the gut. And that's just a fraction of some of these chemicals that are contained inside of an all-natural strawberry. This is actually adapted from a poster by James Kennedy, who has an all-natural banana or an all-natural strawberry posters that highlight like the diversity of the chemicals of the compositions of the foods that we eat. Okay, a lot of information, but basically what we found was if we tried to match a person's microbiome up with the nutrients that they consumed, we couldn't find agreement between nutrients and microbes. But when we considered the food that they used with a complicated kind of mathematical approach, we could pair their microbiome and their food together using this analysis called Procrustes analysis, which I won't go into, but basically we had two sets of points and we ask, do they match up or not? And they do statistically match up when we use the food as the comparator. What's really interesting is, remember, we have samples from each person over the course of 2.5 weeks, and we have all of their dietary information as well. So within a person, we can ask, when this person eats foods that are kind of similar, how does a specific species in their microbiome change in response to that food? And for some foods, we saw that there was consistent changes across the same species across people. So for some foods, we saw specific species bloom in response to similar foods. But for a vast majority of the relationships that we found between foods and microbes, the responses to the foods of specific microbes was personalized. It was variable, and it probably depends on the different communities that a person houses. So microbes might metabolize different foods differently if they have different friends with them. Okay, I promise that's the end of the data dump, and now we're gonna get into some more practical, what should you eat for your microbiome? Okay, mentioned alpha diversity before. Alpha diversity is how many species are there? How many species are in your gut? That's kind of the best marker we have at the moment for what we can use as a marker of like improved microbiome, for example. And so a lot of the research in the nutrition space that's being done has used alpha diversity as the outcome of interest of the things that we're measuring to see if this food has an impact on the gut. We think that if you have low diversity, there's a potential loss for key species that might be essential to keep the equilibrium of the ecosystem. And so we're interested in keeping that diversity high. This is a study that was done called American Gut. So this was a citizen science study. And in American Gut, people voluntarily sent in fecal samples, and they answered survey questions about what they ate and various other lifestyle behaviors. And in return, they got a report that told them which bacteria were in the gut. And what the study found, and they used somewhat arbitrary, a cutoff of 30 plants, they found people who ate more than 30 plants a week had higher diversity in their gut microbiome compared to people who ate less than 10 plants in a week. And so one of the things that we can say with some confidence is that eating more plants improves the diversity of your gut and might be beneficial. The other study that I want to highlight is one that was done recently in 2021. And this study was a randomized controlled trial, and it was a crossover intervention, which is sort of the gold standard we have for studies in this diet microbiome space. It basically means that every person acts as their own control, and so we can trust the data more than if we had studies where each person was just being compared cross-sectionally in a population study. And so for each person in this study, they went on a high-fiber diet for a period of time. And then they had a washout period where they weren't on any specific dietary intervention. And then they went on a high fermented food diet. And when they were on the high fiber diet, some people saw an increase in diversity. And it was probably dependent on which bacterial species were in their gut already. But when they went on the high fermented food diet, everyone saw an increase in diversity. And that increase was also associated with decreased inflammation. And so we have some evidence to suggest that eating fermented foods might increase diversity, and that may be beneficial. Okay, so what's a fermented food? These are growing in popularity. A lot of fermented foods include things like yogurt. That's probably the most commonly consumed type of fermented food here in the US. But also if you're in Minnesota, you probably see sauerkraut with relative frequency. If you go across the street to Korea restaurant, you can get a variety of kimchis and fermented vegetables that are served with your meal. Kombucha is a fermented tea. Kefir is another type of fermented dairy. Miso, fermented soybeans, and then you can make various fermented vegetables. Some pickles are fermented. It's relatively straightforward to make vegetables fermented. Okay, so what does this look like in practice? At the meal level, if I'm trying to increase diversity or fermented foods, like on a per meal or per serving level, what might that look like? So let's say you start with your favorite vegetable. In this case, I'm choosing broccoli. You could have a serving of broccoli. That's great, that's awesome. It meets the dietary guidelines, it's fantastic. I encourage you to do so. Or you could think about your microbiome when you're having that serving of broccoli, and you could turn it into like a slaw, and you could make it into a salad, and you could put some shallots and red onion and almonds and cranberries, and then you could put some buttermilk as a dressing on there, and then throw some mayo and vinegar in just to make it more appealing and flavorful for people in your family. And now you've got, instead of one plant, you have 1, 2, 3, 4, 5 different plants. So that's one way to increase the diversity. You don't have to eat more. It's the same amount of food, but it is higher diversity. And we've got a little serving of a fermented food in there as well. Or we can think about this on like a weekly level. Maybe it's too much work to think, okay, I need to add lots of different things to my food, but I can think about diversifying my diet over the period of a week or a month by mixing in different foods. And so I played around with this idea. I thought it would be way too much work to try and think about diversity every meal. I don't want to do that. Maybe I need to make sure I have flexibility so I can go out with friends to a meal at a restaurant, so I can eat what my family is eating. I don't want to be making my life too hard, right? I need this to be practical. So I'm thinking, okay, one time a day, I think about adding some diversity to my diet. And maybe that looks like having a chickpea salad, making a slaw like I talked about, making a curry, and instead of making it just with one thing, I'm adding a couple other things to it. And if I do this, then I can count up the vegetables or plants that I'm getting over the course of a week. And even while still allowing myself heaps of flexibility to eat out and to go places and to eat what other people are making for me, I can hit like 40 vegetables in a week. And maybe that's not realistic all the time, but if I think about it some of the time, maybe my gut bacteria will benefit from it. So practical recommendations. I want to highlight, really emphasize, the goal here is not perfection. We're not trying to eat some perfect way. We have to be real people living real lives with flexibility to do real things, and our diets should not stop us from living our life. And our gut microbiome is just a piece of the puzzle, right? But if I am thinking about eating for my gut microbiome, I might include a diversity of fruits and vegetables and whole grains and legumes, and this is totally aligned with very generic, sort of generally healthy dietary guidelines in general. It also just happens to be good for your gut microbiome. I want to emphasize this doesn't have to be expensive. We're talking about adding things like canned beans. We can use frozen produce from the freezer section of the grocery store. We can use even canned vegetables sparingly, as long as they're not super high in sodium. So it can be canned foods along with frozen foods, so you don't have to be constantly worrying about having fresh produce. We can build meals around beans and grains and think about focusing on a bean or a grain as the center point of the meal rather than focusing on, say, a beef protein, something like that. And I'm a big fan of beans. I was talking about this in the previous session. Throw some beans in everything. Everything can have some beans. Why not? See what it's like to put beans in your next stew. Maybe you find out that you like it. Same thing with nuts and seeds and dried fruits. It's pretty easy to sprinkle them in and get an extra plant and add a little bit of nutrition for your gut microbiome and add some meal texture as well. And then herbs and spices are filled with polyphenols and other chemicals that we think are probably beneficial for the gut microbiome. And so adding herbs and spices can be great for adding flavor, and your gut would benefit too. And then fermented vegetables, and this says and dairy, I mean fermented dairy, as garnishes or as dressings, as ways to get some fermented food into your diet. So to conclude, my ideas here to conclude is to focus on foods, not on single nutrients, when feeding the gut microbiome. Your microbiome really sees foods, sees the end products of your digestion of food, and is less worried about was it high in carbohydrate, was it high in fat? That seems less important than that there was a diversity. If you do want to pick a nutrient to track and you're thinking about your gut microbiome, the nutrient that I would pick would be fiber. And often if you track fiber, other nutrients just fall into place. So we're trying to get around 28 grams to 35 grams of fiber a day. Beyond that, it doesn't seem to help the gut microbiome that much. So most Americans, for reference, get about 10, maybe 12, sometimes 15 grams of fiber per day. So 28 grams is aligned with the dietary guidelines, and most people don't come anywhere near it. If you could come close to that, that would be fantastic. Pick a diversity of plant foods, including fruits, vegetables, whole grains, legumes, and these might improve microbiome diversity. And then we have some evidence that fermented plants and fermented dairy can also improve microbiome diversity and may reduce inflammation. And I want to emphasize that this is an emerging area, and we don't yet fully understand how foods influence the microbiome or health and cancer. A lot of this research is still yet to be done, is being developed as we speak. We're doing lots of pilot studies to try to understand how preparing the microbiome before cancer treatment or improving diet after cancer treatment may help to improve outcomes, but we don't yet have answers that are really solid. So with that, I will open for some questions. If you want to ask a question and you're unable to today, my email address is here, and my Twitter handle is below.
Thank you, Dr. Johnson. We do have a few minutes for questions before we break for a lunch. And show of hands if you have questions. I'll come over to you with the microphone.
How big of a difference it is eating fresh versus, for example, spinach, cooked spinach versus fresh spinach?
Yeah, that's a great question. We don't know the answer yet, cooked versus fresh. I could imagine that there may be some differences, but they're probably not enormous. There are some ways that food changes. In the previous session, we were talking about resistant starch. So for example, if you cook a grain like rice or pasta and then you cool it down, there's a production of a compound called resistant starch. And that can change, is metabolized differently by you and by your gut microbiome. For something like cooked versus fresh spinach, I don't think that we know. Part of the challenge in this space is that it's really hard to get you to tell us what you ate when we're doing this research. It's even harder to get information about the preparation of the food that you ate. So it's often difficult to tease down into that kind of level of detail. So for the most part, we don't know. Until we do a randomized control trial of cooked versus fresh spinach, we won't have an answer. And it's really challenging to do that for every type of food, for every type of preparation. So at this point, I can't say that there's a difference, but my guess would be there's probably not much of a difference. You get more food if you get more spinach per serving if you cook it, yeah.
Alkalinic versus acidic foods?
Again, I don't know the answer to that. Everything that enters your stomach is going to be acidified by the stomach acid that you have. And then the pH of the food as it moves through your small intestine is changed again until it reaches your gut. Whether or not the pH of the food that you ate has an impact on the distal gut microbiome, we don't know. Yeah.
I had a question before we break. I'll head over there. Is water a nutrient for the microbiome? And if yes, how and what way?
Yeah. Water is, again, one of those things that's quite hard to measure in these research studies. No matter how much I tell my participants, I care about how much water you drink, please tell me, they often don't record the water that they drink. So I don't always trust the data that I have on water consumption. However, there is really interesting research about what would it call, like fecal composition, or like, have you heard of the Bristol stool scale? So whether or not your fecal pellets are like hard rabbit poos versus if it's like fully liquid diarrhea, that's the Bristol stool scale. We can ask people to tell us how firm or liquid was their stool. And that has a strong relationship with the bacterial composition. So people who have a more constipation-like microbiome or constipation-like stool have a different microbiome to people who have a more liquid stool. And some of that might be because transit time is wildly different for both of those people, where the constipated individual, that community is sitting there for much longer and it's changing more because the dynamics are different when it's sitting there in contact with the food and with the host for a longer period of time than the very liquid stool. And that relationship may be related to how much water a person is drinking along with the fiber. If you eat a lot of fiber without taking in water, you can lead to this environment that's more constipation-like. So if you're going to increase your fiber and you haven't previously done that, anytime you increase fiber, you should be adding water to the mix, because fiber can gum you up and adding water can help prevent that. So it's kind of that. Does that answer your question? Yeah.
That is a nice way to move into, should we be taking fiber supplements if we have a hard time getting that in our diet?
Yeah, that's an excellent question. Should you be taking a fiber supplement? And I think you can try it and see. The challenge with a fiber supplement is all one thing. And so we do think that having lots of different types of fibers is probably a good thing in terms of allowing the different communities. Like if you think about feeding a specific bacteria, you would want it to have the food that it likes. But since you've got lots of different bacteria, you probably need lots of different types of foods for all of those bacteria to grow and have like a thriving ecological community. And if you throw just a lot of one fiber, you might see that improves for some people who happen to have a bacteria that really blooms in response to that specific fiber type. But we don't yet know like if that's as good or better or worse than getting a whole lot of fiber from the foods that you eat. So my suspicion would be it's probably best to get it from your food, but if you're really struggling and you want to try a fiber supplement, absolutely give it a go, but remember to drink the water with it because it's a lot of fiber in one go. And if you find after a couple of weeks it's not doing anything for you or you don't like the effects, you could change.
Hi, since this is a newer field, you said, what sorts of studies are you doing what do you want to learn first, sort of thing? And do you need participants locally for any studies you're doing? I'd like to help.
Yeah, we have so many studies going on. I don't have a single place for you to go to look for the repository of studies that are available. Right now, we're doing pilot studies looking at if the microbiome is changing or if we can manipulate that around the time of cancer surgeries for colon cancer. That would be like the #1 study that I'm working on at the moment, but we should be wrapping up enrollment this month. So I would suspect most people are not eligible for that one. Sporadically, we have other studies, but I can't think of any that are really open at the moment. It would be great to get some sort of cancer survivor cohort with microbiome samples, so maybe Anne wants to make that happen. But we don't have anything right now that people in this room could just volunteer for. Yeah.
This was fantastic. Thank you so much. Your presentation made me curious about the trend of meal prepping and eating the same food for lunch every day for a whole week. And thinking about that in the light of going for diversity and 30 plants, I suppose you could make those very diverse meals, but then you're eating those very diverse meals for a week at a time. Is there any research or area of inquiry that's in that space?
Yeah, nobody has done that research yet. I think it's an excellent question. It's one of the things that comes up when we start to try to think about what is a diverse diet. There's a lot of different ways that you can envision that. You can think about it like each day you have a diversity of foods versus over the course of a week or a course of your life you eat a diverse and changing diet. Mostly the field of nutrition has not thought about diversity. Generally, nutrition has really focused on macro and micronutrients. It's thought about excluding entire food groups, but this idea of diversity is baked into a lot of the measures we currently have for what is a high quality diet, but it's typically not measured specifically or used as a way to look at health outcomes, in part because of what you've identified, that it's difficult to articulate what component of diversity are you actually interested in. Potentially, there are different ways that you implement diversity could have different outcomes. So, to be determined? Excellent question.